I think that's important. How is everybody? We all good? Freezing to death?
Yeah.
Actually, Julia, you look like really kind of not freezing to death. Whereabouts are you?
No, I'm in the Blue Mountains. But
why are you wearing a t-shirt, you crazy person?
I went for a run like a crazy person to get some of the,
yeah, no, that's
out, so to speak. And I've got this sun coming right through here. I've just been sitting studying. It's really nice.
Oh, that's lovely. I was a mountains girl once upon a time. I was a Bullaburran.
Wentworth Falls.
Oh, there you go. So we were right next door. Yeah. So, well, it's bloody freezing here, got to say. So I'm rugged up and I've got thermals. I've got everything going on today. But Bathurst is that bone cold. It just gets into your atoms and it won't leave. So excuse me for looking like an Eskimo, but that's the way it's going to be today. So anyway, today we're doing last week's work on blood, just making sure that it's all nice and clear, and it's pretty simple. I think this one's not going to stretch you too much. So let's get going without further ado. Okay, so easy beasy. Obviously, I say this every week, so I'm not going to say it this week. You know what learning objectives are and you know what you're supposed to do with them. End of story. Okay, let's move on because I'm not going to read them all out to you because you're not reading challenged. Here we go. Hopefully 1, 2, and 3, this should be bang, 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 easy, easy. So normal blood pH, go. Which one?
B.
B, absolutely, 100%. And we on either side of that, of course, is acidosis and alkalosis, and it's really fast. So it's a very narrow pH range that we've got to stay within. Okay, oxygen is carried around by something. Which one?
C.
Yep. C. Definitely not granulocytes. Cool. So what I'm hoping is that not just is everyone jumping to the right conclusion, but also going, why is the other stuff wrong? So neutrophil, no, that's not what that does. Platelet, oh, definitely not. Okay, the process by which we stop bleeding and create nice large clots is which one?
Homeostasis?
Not quite. Nearly.
D.
Yes, hemostasis. So that, yeah, watch the spelling. Yeah, so 2 different processes. Homeostasis, literally meaning keeping our entire system at whatever pH it needs to be, at whatever pressure it needs to be, whatever. That's all of the keeping your system at one place. Hemostasis is specifically blood.
Okay, cool. Thank you.
That's right. And it is tricky because, as someone asked me on Monday, and it was a good question, because on another part of these slides, I've actually written hemostasis as HAEM, ostasis. And that's confusing people too, because there's more vowels and they're going, but it looks even more like homeo. Does everyone understand what goes on with that whole HAE thing? We always used to write esophagus as OE as well, like an oesophagus, and HAE, hemostasis. It's from the Latin roots, and because language evolves and changes, they've kind of dropped out the Latin spellings and made it more kind of universal. Right, so oesophagus.
I did not know that.
Yeah. So oesophagus is no longer an oesophagus; it's now just an E. It's an esophagus. End of story. So yeah, it's just the Latin stuff that's starting to slide a little bit because it is a dead language, and although the roots are there, it's more accessible if it's easier to spell. Okay, so that's reasonably easy. Everyone went 1, 2, 3. Good. Who is our universal donor in bloods?
O.
Yes. Although this question is not quite strict enough. What is missing from that list? We've got all the different types, but what are we missing?
The positive and negative.
The positives and negatives, absolutely. So is it O positive or O negative that is the universal donor?
O negative?
Negative.
Yeah, so O positive is the blood that everybody wants because it's 37% of the population has O positive blood. So we want lots of it in there in the banks so that we can do it. But O negative is the universal donor. Why? Because it doesn't have something. What does the positive and negative tell us about that person's blood?
It doesn't have the antibodies against A and B?
No, not quite. So the positives and negatives is to do with the rhesus factor or the RH
Oh,
factor. Yeah.
Yeah.
So if you have O negative blood, you literally have no antigens, nothing going on. Yep. So it's beautiful. It's basically clean, wonderful, pure, and particularly useful if any of you end up working in pediatrics. O negative is the blood that we want to give to newborns and things when they need blood. Yeah. Okay.
Yep.
Hopefully the next one is really simple. The 2 upper chambers followed by the 2 lower chambers. Which one is it?
B.
Yep.
Oh, yes.
Is everyone 100% on that? Because that should be a really easy one, I should think. Atria ventricles, we know that. Yep. Okay, cool. All righty. Oh, I've just got a quick question for you on our group O, actually, before I jump off that, because it's a really interesting one. And it's kind of your bread and butter, and you really need to know blood well, obviously. So if I'm O, which I am, for instance, and at least a third of you in the room will be O as well, because it's 37% of the population, I don't have antigens on my red blood cells. Yep. But I do have antibodies. Yeah. I've got anti-A and I've got anti-B. So how does that make me a universal donor? Aren't my antibodies going to attack other people's blood? This always confused me. The reason I ask is because when I was learning this, I went, I don't understand. I've got the antibodies though. So what, how come, what? Has anyone got an answer for me? It's tricky, isn't it? Because you go, hang on, how can it be a universe? If I've got anti-A and anti-B antibodies in my plasma, how do I give my blood to a group A? It's going to attack the blood cells. Or a group B, it's going to attack their blood cells, or an AB. This did my head in forever until someone explained it to me. And I'm like, it's so simple, actually. Does anyone know the answer to that off the top of their head? It's probably not in your lecture notes or anything. It's because we don't actually give the whole blood. We only give the red blood cell packed blood. We actually take the plasma out. So when you say universal donor, we're not actually giving the whole blood. We're giving packed cell volume only. Yep. So that makes, they literally take my antibodies out before they give my blood to somebody else. So there aren't any of that going on. It's literally just the red blood cells. Yep. Which I,
Do we need to know that, Catherine?
For your life, yes. For your for your tests and exams, no. It's just I wanted to land on it because it confused the hell out of me when I was learning this, and it was just one of those schisms that didn't make sense. And I went, okay, I need this explained. Yeah, Lara.
I thought I'd just ask this because when I came onto Zoom, the test, like the not test, the little questions and stuff had already kind of started. So is this, are we reflecting back on things that we'll be doing in the test that's opened today? Or
Yes.
is this for, okay, I thought so, because it looked like a bit further away from like topic 8 necessarily. And I thought, oh, this, yeah, okay,
Yeah.
that's good to know.
Yeah, this is last week's material. So
Yeah.
we're always covering it the week later. Yeah.
Yeah.
Yeah. So yeah, I know that's so yeah, good question, Angela. But no, you're not going to be tested on any of that. But it was a schism in my mind when I was learning it. I thought I'm going to explain it and make sure that it's clear for you guys. Julia.
Yeah, sorry. Well, if you're only giving the actual red blood cells instead of the plasma, then
Yeah.
how come like you can't cross A and B? I think I've got
Oh no,
confused.
it's only with O that we do that. So
Oh,
O
right.
yeah, O is given as packed cells. Everything else is
Okay.
actually given as whole blood.
Okay, thanks.
Yeah. Does it make sense now? I was like, yeah, okay, yeah, good question. Yeah, it's all those little schisms that stop you really grabbing the learning. It's like that, but that doesn't make sense. Oh, now it does. Yeah, good. All right.
Okay.
Now this one's pretty simple. Listing the components of blood. I did put a picture up so that you've got a hint. Can anyone do it off the top of their heads? What's in blood?
Nutrients, oxygen, and like the blood cells.
Yep.
And
Cool.
like
So
carbon dioxide.
yep, cool. So let's go into that. So we've got two very different parts of blood, really, don't we? We've got the formed elements And then we've got the juice, right? Or plasma, for want of a bit more scientific. So what are the forms? So we've already got, so you gave me red blood cells, sweet. What other formed elements do we have in our blood?
So the red blood cells, white blood cells, and the platelets.
Absolutely. Yep, formed elements. Very good. And in terms of then you go into your plasma. What's in plasma? And so what have you got? Lara, you gave me nutrients and you gave me oxygen and you gave me, what else was there in that list?
Waste, like waste components.
Yeah, yep, absolutely. Anyone else got anything else for that list? What's it made of?
Water?
Mostly? Yeah, that's a pretty important part because it's most of it. Let's have a look. I'll show you in the next bit. Okay, so with your formed elements, you must make sure that you just separate it in your head. I've got to talk about my formed elements and then I've got to talk about the plasma. Now, as we see, 90% water, pretty important, mention this, but all of these different salts, all of these different proteins, which is actually important when we start talking about clotting. So remember, we've got all those marvelous little fibrin proteins and things that actually help us out when we get to the clots. And then, as we said, nutrients, waste, gases, hormones, tons of other things. However, you don't have to remember everything in that table because the list that's given to you on the right is pretty much where you're expected to remember. So just electrolytes, nutrients, waste, gases, hormones. Yep. But for greater understanding, I think it's really important to, for instance, look at the plasma proteins. As we said, things like fibrinogen, particularly in this topic, super, super important because we're talking about hemostasis and clotting. So you need to know, it's like, oh yeah, those elements are always floating around in plasma, but when I get that compromised cell wall, that's when they actually come into play. Other than that, they're just floating around in plasma. Yeah. Okay, cool. Let's keep going. No, I didn't want to do that. Yes, I want to do here. Okay. Did I just go forward 2 or 1? What did I do? No, I did 1. Okay. Awesome. Okay, definitions of viscosity and osmolarity only as it pertains to blood. So let's start with Mr. Viscosity. Someone give me a nice, simple, not too jargony understanding of what we're talking about when we're talking about blood viscosity.
The density of plasma to formed elements.
Yes, absolutely. So brilliant, jumping straight to hematocrit. Good answer. Excellent. But if you're explaining it to a kid, viscosity is what? Thickness. Yeah, stickiness. Like syrup, or hopefully not. Hopefully your blood is not like syrup, but generally viscosity is thickness. So what happens if I have increased viscosity in my blood beyond the ideal parameters? What's going to happen? Is it going to move easily? Or is it going to move slowly?
Slow.
Yep, because it's literally going to be sticking to the walls. Yep. Okay, so increased blood viscosity. What causes increased blood viscosity? What elements have we got too many of in our blood when blood is too thick? Does anyone know that?
Is it red blood cells?
It is red blood cells, yeah. And it generally happens because of a lack of oxygen. Yep, so this happens in mountain climbers all the time, right? So their body perceives it's not getting enough oxygen and it pumps out a whole bunch more red blood cells in order to get more oxygen circulating. Another one is heavy smokers or smokers in general, actually. Yep. Because again, your body is perceiving not enough oxygen and it's going to pump out more red blood cells than it needs. You end up with thick blood. Yep. So not a good idea to take up smoking. If you haven't, don't think about it. Okay. That's viscosity, thickness. And yes, it sticks to blood vessel walls and means that we have all sorts of problems there with pressure and buildup and oxygen delivery to tissues and all that kind of thing. All right. What about the next one, the scary chemistry word, the osmolarity? What is blood osmolarity?
That's how much solute particles are in the solvent?
Yes, thank you, chemistry, Bronson. Brilliant. Okay, we used words like solvent and solute. Yay, good. Yes. Okay. So yeah, the dissolved elements in the solute. Sorry. So why is that important when we get to tissue level? Why is it important to have exactly the right balance of solutes in our blood at any given time?
Because if there's too many solutes, then there's too much viscosity of the blood that would reduce the blood flow.
Partially, although that it is sort of a secondary consideration. Possibly, generally, we're talking about so small it's not really going to affect the flow, because if you remember, you've got like your blood is literally 90% water. So you would have to have an incredible increase in these solutes to really affect viscosity. It's the red blood cells that mainly do that because of size. But let's think about movement of ions in and out of cells. So we know about osmosis. Do we not? Yes, we know that water follows salt, etc. So what happens if I have an enormous amount of salt in my blood? What's going to happen to me? Where am I going to pull water from to try and balance it?
The cells?
Yeah. And everywhere else. Yeah, anything I can get. So basically we're upsetting balance. Yep. Because nearly so many things. Think about neurons. Think about heart conductivity. We're pulling ions all the time in various different places, and they need to be in a particular concentration for things to work. So if I have a huge amount of sodium in my blood, for instance, the sodium channels in the heart's electrical system are always open. Yep. So if they get a massive inrush of sodium all the time, that is going to make my heart beat much faster. It's going to make it work too hard. Yep. You need to keep the balance, right? So a lot of stuff is about movement of ions across membranes. Yep. So we don't want to have those ions out of balance. Is that clear enough with everybody? So viscosity is completely much more to do with your red blood cells, because just in terms of percentages of what's in there. And then we've got 90% water, and then we've got our solutes and different substances actually dissolved in that water. Yep. But they've got to be in good balance. All right, let's move on. Oh, okay. So I put in some slides with the answers. How's our viscosity? Yep. Lovely. And then our osmolarity there. Yeah, so movement in and out of your capillaries. Pretty important. Okay. All right. And you don't have to remember, I don't think, no one's ever going to ask you to write out that osmolarity means the amount of solutes per litre of solvent. That's very chemistry. But it is important to remember that. Okay. And we just talked about that. Increased blood pressure, volume, and more strain on the heart. Yep. Okay. Now, this one is not just knowing about the formation of blood cells, but just being able to play language games, really. You've got to find, even if you didn't know the answer, you could find the answer, because you just got to go logically with the story. So where do we start? What's number one here for the formation of a red blood cell? Have a quick look and see if you can find it. Right, anyone got it? Number one, step one.
I'm going to say F.
You so could be right. What was your second choice?
D.
Yeah. I love the fact that you said that, actually, because
Sure, because it could have gone either way.
Yes, you're absolutely. I was going to say, remembering, of course, that this is cyclic. So actually 1 could be 6 and 6 could be 1. So there is that. Yes, so it's either F or D, right? Let's go with D and just put F at the end. But you're so right. You're absolutely right. So if D is 1, what's 2? Follow the language. We've just said EPO. What's the next sentence that mentions it?
E.
Yeah. See, sometimes you
Hmm.
don't even have to know this process. You just got to follow the language. Yep. So we're good. All right. So the kidney's just made EPO. I'm going to jump there. EPO stimulates the cells in your red bone marrow to create some new erythrocytes. Then we jump to what?
A.
Yep, absolutely. Then?
C.
Yes. And finally, we only have one left, but we have two left because, then to
E.
E, and then obviously to F or back again. Yep. Okay, is that nice and clear for everybody? We just got to jump from kidneys and EPO and then into marrow and then into circulation. And then, as they die off and we get rid of them, then we need to create more. So it's a nice little cyclic thing. All right. Now, the other end of it, of course, is that they do have a limited life cycle. Off the top of your head, does anyone know what the life cycle of a red blood cell is approximately?
100 to 120 days.
Yes. Yep. And then we need to break them down because they stop working. Like all of us when we age, you stop working. Okay. It's me being mean. I'm just having a day with old things today. There's all these questions in the paramedics lab book about the effects of aging on stuff, and I feel personally attacked. Anywho, I don't have any problems, thanks. I'm just as sharp as I always was. All right, so let's recycle some blood cells and see what we can talk about. So obviously our liver and our spleen would be our red blood cell graveyards, right? That's where they go to die. It's really nice. And we've got macrophages in there that do that for us. Pretty much their job is to pull everything apart and get it recycled where possible. Okay, so what is the first level of that talking about? The first, underneath the first arrow, where you've got globin on one side and heme on the other. Someone talk to me about what globin and heme's all about. What's going on there?
The globin that's the protein that I think breaks down into amino acids.
Yes, absolutely. And heme?
The iron part.
Yeah.
The bilirubin or?
Yes, that's right. That does end up at bilirubin. Yeah. So it's just literally the two parts of the hemoglobin molecule, isn't it? Taken in apart. One side is the heme, which is the heme molecule, which is, as you say, the iron carrying molecule. And one is our nice big chains of amino acids that we can chop up and use again. So we need to keep those. Okay. So then the next one down, exactly as you just said, we are breaking down our globin into individual amino acids. That's great. Put them back in circulation. We can build some stuff with that. That's building blocks. And then out of the heme molecule, we have our iron, and we have, as you just said, bilirubin is the other output of that heme molecule breaking down. We want to keep the iron, do we not? Yes, we'd like to keep the iron. Good. So we keep the iron, and the iron generally is going back to the red bone marrow. Yep. The globin that's been broken down into individual amino acids is, as we just said, is going to be built back into something else. Yep, whatever we need it for. And the bilirubin, on the other hand, what do we want to do with that? Do we want to keep it?
No.
No, we chuck it. Where do we chuck it? Urinary or fecal?
I think urinary.
Yep. Yeah, and actually also fecal, to be honest, because you shouldn't have too much bilirubin in your urine. If you do, it's a problem. So it's also fecal. But yeah, it's a charming compound that we want to get rid of. It's very smelly, I have to say. Bilirubin. Don't ever open a test tube that says that it's got bilirubin in it. It's disgusting. They do that to you in first year when we did in the olden days when I did my degrees. Like, smell this, guys. Oh, I hate you. Okay, so breakdown of red blood cells. And literally, that's just the word version. Okay, so the one thing you don't need to know on there, that's your life cycle of red blood cells, just put in very, very simple points. important bit down the bottom there on the right hand side, you were given this little flow chart. You don't need to know about protoporphyrin in between. Just remember that heme is largely broken down into iron, which is reused, and bilirubin, which is excreted. Yep. Okay. Ignore the protoporphyrin. There's about a bazillion other steps in the middle there. Okay. Now I'm hoping this one's going to be simple. Now Julia's feeling it.
Yeah.
Oh yeah, it is cold. Okay. We need to describe the pulmonary and systemic circuits. So again, I always like to say, imagine I'm 10 or, you know, I'm not a scientist. Explain it to me so it's easy to understand. And somebody can do the pulmonary and somebody can do systemic. I don't mind. You can take half each. I really think it should be someone who's not Bronson, Lara, Angela, or Julia. Does anyone else want to jump in and tell me what they understand by the, well, you know, it's a good idea too, because if you get it wrong, I can correct you and then you'll know.
Your pulmonary is like your lungs and stuff. If I was explaining it to a kid, it's your lungs.
Thank you. Yes. So, and specifically from what to lungs? Heart. Yeah. Yeah, because so when we're talking about these circuits, this is circuit from heart to lungs, and systemic, obviously, heart to the everything else. Yeah. All right. So in the pulmonary, now this is where we get to the who's been studying their stuff. When you get to your ventricles, right? So let's just imagine our blood's done all its thing, it's come in, and we get to the ventricles. Which pipe takes everything to the systemic circuit? What's the first pipe that oxygenated blood hits before it goes out to the rest of the system? The what?
The aorta.
Aorta, absolutely. Do you remember what the name of the valve is in the aorta for extra points?
Oh, it could be, but I could be completely wrong because it's been a long time since I did the heart anatomy. I think it might be the mitral or the tricuspid. I could be wrong.
It's semilunar.
Oh,
that's okay. That's all good. I was going to say, just think of the moon. It's going up towards the moon. Okay, good. What's taking the blood from the heart to the lungs? Which pipe? Sorry?
Can I answer? Or?
Yes, you can, Angela. I'm just being, I was trying to get other people, but, jump in. Yep.
I'm happy to just be quiet, but
no.
I don't want to be that person. So
it's okay, because you're also teaching while you're learning, so it's a good thing. It's fine. So which pipe?
I could be wrong. Is it the pulmonary arteries?
Yes. What's weird about that, guys? So what generally in pictures, right? We have arteries are red and veins are blue, right? Generally, arteries carry oxygenated blood. Generally, veins carry deoxygenated blood. Not from the heart. All right. So that's one place where it's switched. Okay. So the actual definition of artery and vein, this is kind of important. Artery takes something from the heart, and vein takes something to the heart. It actually has absolutely nothing to do with the oxygenation of it at all. Okay, so it's just artery from vein to. Yep. So for instance, as Angela just said, your pulmonary artery takes blood from the heart, but it's not oxygenated blood. Right? It's deoxygenated blood. It's going to the lungs to get oxygenated. When it comes back to the heart, it comes back through the pulmonary vein, which is not blue. It's bright red because it's been oxygenated by the lungs. So in the heart situation, it's actually the opposite to most of the rest of your body. Generally, you'd be okay saying most arteries are oxygenated blood and most veins are deoxygenated, but not all. So always remember the heart is flipped, slightly different up there. Okay, So we've got our heart, we did our major pipes. We're not going to go into the branches because that would be insane. But that's basically your pulmonary and systemic circuit. Quick question before we jump off it though. Which circuit has the highest pressure? Do you think? Which one's got to go further?
Systemic.
Systemic. Yeah, absolutely. How far do you reckon? Think about it. Here's your heart sitting here. And how far away are my lungs, do you suppose, from my heart? About 2 centimeters. Not a lot of pressure needed to get the blood from one place to the other. Right? So your pulmonary circuit is far, far lower pressure than the rest of the systemic circuit because it's got to go, everywhere. It's big. All right. Is that fairly clear, I think, to everybody? Pulmonary and systemic?
Yeah.
Yeah, cool. All right. So just on this one, just quickly, as we said, high pressure in systemic, lower pressure in pulmonary, and you don't really need to worry too much about anything else on that list except for making sure that, one's lungs only and one's everything. Okay. Now, as we were talking about fibrin and fibrinogen earlier on as components of blood, this is where the thing came up on Monday because I wrote hemostasis in the old Latin way instead of just with an E. So this confused people. Okay, I like it like this though. I like the Latin thing. It I don't know, appeals to me language wise. All right. So hemostasis. What is hemostasis in a simple response?
Blood clotting.
Blood clotting. Yeah. What does it actually? What do the two Latins actually mean there? Do you know? Hemo meaning
Blood.
Blood. Stasis meaning
Stop.
Yeah. Yep. Blood stop. Precisely. So how do we stop blood from leaking out where it's not supposed to go? Good. All right. So what is this process of hemostasis and where do we start? So we've defined it. Good. Where does it start? Do we have to assault a blood vessel first, don't we?
A vascular spasm occurs after we have the assault of the blood vessel.
Yeah, right. So we do something nasty to misdraw a blood vessel, and then like everybody that gets hurt, the first thing he does is go, "Oi, jeez!" Right? Vasoconstriction, muscle spasm. Yep, of the smooth muscle. Okay, which achieves what? What is the vessel constricting achieve? What's the point?
To minimise the loss of blood.
Yeah, absolutely. It's what? Shut down. Everything shuts down. All right. So we've got vasoconstriction. That's number one. What's number two? What happens after that?
The platelet plug forms.
Yes. Now, for extra points, why? Why does a platelet plug suddenly form? Well, how do the platelets that are normally floating around suddenly go where they're supposed to go? Are they psychic or was there something else? So this, yeah, no, go. Yep.
I think. Yeah, sorry. Want to go?
You go. You go.
I think the damaged blood vessel releases collagen, and then
collagen is exposed. Yeah.
Collagen's exposed.
Yes,
and then
that's right.
Adhere to it so they move towards it and stick to that area.
Yes, absolutely. So as soon as collagen is exposed, so collagen is like platelet heroin. They get very excited and they become sticky. They actually change in nature. So normally they're free floating around in the plasma, just doing their thing, not bothering anybody. As soon as collagen is exposed, it's literally red rag to a bull or honey to a bear. Platelets get terribly excited, want to get as close to that collagen as they possibly can and become sticky and start to stick to each other and the site of the blood vessel injury as well. Good. So that's platelets. That's first, but it's only part of the story. What's next? So when we're talking about hemostasis, there's one really important thing we've got to mention. Actually, two. One is one of the formed elements we've talked about already.
The coagulation event occurs.
The coagulation cascade, which luckily you guys don't have to remember. Thank God. All those clotting factors are evil. However, you do have to mention that there are clotting factors. Yep. So clotting factors that are activated, if you want to use the word cascade, awesome, in a cascade. And those clotting factors are going to make sure what happens next. So on the picture that I've given you, you can see platelets, right? So the groovy little starfish, that'd be the platelets. What are all the threads?
The fibrin.
Yes, absolutely. Fibrin. Good. And fibrin was floating around in our plasma before, remember, as one of our proteins that exists all the time. Yep. And what does the fibrin sort of do? What's its function in this clotting process?
It forms a web, like
Yeah.
A knitting kind of thing.
Yeah, netting, webbing, exactly. And what is it trapping? You can see it on the picture. Red blood cells. Yep. So that gives us a nice big clot, doesn't it? That's pretty much the process. Let's have a look and see if I've got that written down for you. Yeah. So vasoconstriction, platelets get activated. The mysterious coagulation cascade, which we don't need to worry about except that it happens. So yay. And the big one that comes out of that, though, in terms of the clotting factor, is our old mate thrombin. Thrombin is worth remembering. Okay, thrombin is important because thrombin is what makes fibrin do what it does on the blood clot and actually turn into that web. Has anyone ever, it's not as common now, but there's actually a condition called a thrombosis. Yeah, which is a clot. Yeah, so thrombin, thrombosis. All right.
I actually saw that on RPA the other night. They were talking about it.
Yeah. Yeah, it's well, my grandmother used to get them all the time. She used to fly a lot. She used to get lots of DVT, deep vein thrombosis. So she had really, really funky stockings. They were so sexy. It's pressurized stockings to stop the DVTs. All right, so you've got a primary hemostasis there and a secondary hemostasis. You don't need to worry too much about those distinctions, but it might help you compartmentalize. In primary, it's the constriction and the platelets and the collagen doing all their thing. And in secondary, that's when we have all the other factors coming in to help and seal up the plug and make it really, really strong. Yep. To me, it helps to compartmentalize things a little bit when I'm trying to remember. Okay. And when we get to the end of that, obviously the great news is that we don't want a clot sitting there forever. So then we have our process of fibrinolysis where we break it down again once it's done its job and recycle. Okay. Oh, now I was wondering why I put this up again. I am not going to sit here and watch you fill out a table. But are there any questions? Have a quick look at it on the document. Is there anything that's confusing there? Yes, Lara.
This is naughty because it's not exactly related to this, but I have a question and I don't want to forget it. But
Okay.
it was related to what you were just talking about with blood pressure changes, how you said it was higher as it went away from the body. Is that correct, what you said? When you
The
have
blood pressure is higher.
it's higher, like when you said systemically rather than pulmonary?
Okay. Broadly, your systemic blood pressure is higher than your pulmonary circuit because it
Okay.
needs to be. But having said that, blood pressure is always highest at the heart and steadily decreases as it goes into extremities.
That's what was confusing me because when I was looking at a PowerPoint from the lecture, that's how I make my notes. And it just had this picture. And at the highest, it was basically the highest of your systolic and your diastolic. That's your reading. And it showed how it went down with the corresponding like vessels and all that.
does.
Like the end of your peripherals.
That's right.
And then to me, it said, okay, well, the closer it is to the heart, the higher the pressure because it's got to send it down to the body. So I was just a bit confused with that image. And then how you said like it's higher in the systemic. So I just want to double check for my own understanding.
No, that's fine. And it's a good question. So does that make sense now when I say overall, the systemic blood pressure is higher because it needs to be from the simple point of view that it's got to pump further. So it's got to have a higher initial pressure than because it doesn't have to go as fast to the lungs in the pulmonary circuit. But having said that, yeah, the pressure is very high here. And by the time it gets down to my feet, it's not at all. Yeah.
All right.
And as you say, with the size of the vessels as well, as they lessen, of course, that's going to have an effect as well. Okay, so are we okay to fill out the table by ourselves? Is there anything on there that's confusing? Is the RH factor okay with everybody? That tends to be a tripping point.
Can we go through that, please, Catherine?
Yeah, because it's tricky, and it's sort of because you learn the old ABO thing, and then you go, oh, there's another one. Why can't we call it all the one thing? Yeah. So the RH factor, or rhesus as it used to be called, this is another antigen actually on your red blood cells. Okay, so it's not some magical thing that floats around. It is another antigen with, you know, the associated antibody. So the problem with RH is that if you have the RH antigen on your red blood cells, then you've got the antibody as well. All right. If you don't have the antigen, if you don't have any RH antigen on your red blood cells, then you don't have an antibody for it either. The only problem that ever occurs with this is when two opposites have a baby. Okay, so the way that this works is really simple. So if your mum is RH negative, dad is RH positive. Right? So, but at this point, they're not sharing bloodstreams. It's fine, right? There's nothing that's going to stop them doing anything. It's all lovely. So she gets pregnant. That's fine. Baby grows fine. Everything's lovely. And we get to delivery. So the pregnancy is absolutely fine because there's no crossing of bloodstreams here, right? So we have an independent bloodstream going on. Baby's blood and mum's blood are not mixing, so it should be absolutely fine. During childbirth, though, things can rip, things can tear, compromises can happen where those bloodstreams actually do end up mixing. Then what happens is that that mother's blood is now going to start making antibodies because it's had a foreign antigen introduced. Yep. So next time she has a baby with said man, her own body is going to try and attack the fetus's blood cells. It's going to recognize it as a foreigner, as an invader. Right? So it's only when bloodstreams are crossed. It shouldn't really happen, but it can, and it sometimes does. So this is why every single mother with Rh negative, we always give them RhoGAM to make sure that that antibody reaction doesn't happen. Just in case. It sometimes doesn't happen. It's an insurance policy to make sure that.
Ask Catherine, before that was introduced, would those two people just sort of not, like they might have one child if things go well, but then they wouldn't be able to breed essentially? And
yeah, often, and this was really tragic.
Biological thing to not make a species, continue existing if we hadn't.
Yeah, no, I know, and it's this often was the case. People had one perfectly healthy baby, and then there was a series of miscarriages.
Yeah,
and literally it was just put down to bad luck.
Yeah,
like, well, that's terrible, obviously, but they had their one, so there was something great about that. But it wasn't until we started looking at these rhesus monkeys in the 60s and 70s that we actually figured out what's going on. So a lot of heartache and a lot of time for science to catch up, really. But science is not infallible. We screw it up all the time. Okay, now I think this is your last question. Someone who's got the paper in front of them. I haven't got the actual piece of paper in front of me. Is that the last one? Yeah. Okay. So now we've got these valves in the heart, obviously, but a number of them. You can use their proper names, or you can actually just call them right and left atrioventricular valves, because that's also right. It's correct. You could do the mitral and you could do the bicuspid. It doesn't matter. Either way, there's valves between the atria and the ventricles. Yes. And there's valves going up into the aorta. Yes, right. What happens if somebody has leaky valves? So let's answer the two questions that I've actually put up there as prompts for starters. So if I've got a leaky right atrioventricular valve, what is happening to the blood?
It's falling back into the atrium or the ventricle, wherever it came from. It's falling back into it, like you're not able to circulate properly.
Yeah,
because you're not actually being able to pump the blood properly through the rest of the body, starting at the heart.
Yep, absolutely. So if I've got blood going back in the wrong direction, so for instance, I've got the valve is opened and then it's closed again. So I filled my ventricle, but now it's going bleh, wobble, and I've got some backflow into the atria. Yeah. So why is that a problem? Because it is just going to go back down eventually. Like, it's not like it's going to explode or anything like that. It's what is that volume of blood being there doing to the ventricular, I might just give you a big hint, pressure? Is it changing the pressure in those two different areas? Yeah. And that's the big thing with blood flow. So blood flow is always going to go from a place of high pressure to a place of low pressure. That's kind of how it works. Yep. So first of all, atria fills, because it's full, it is highly pressurized. That opens the valve to go, the blood will go to the ventricle, which has no pressure until there's a whole lot of blood in it, right? Then we have massive pressure in the ventricle, which beats the pressure in the aorta, and that's where it goes next. It keeps on going to the place where there's lower pressure. Okay, so this is how it flows. When it goes backwards, we're affecting that pressure, right? So we are affecting how much and how effectively the heart is going to get the blood out to the lungs or to the rest of the body. Right? The pressure is affected. Does that make sense?
So just to double check, is the systolic blood pressure, is that the pressure of the ventricle squeezing blood out of it? Is that correct?
That's right.
So if the ventricle no longer has the blood and it's kind of going back in the opposite direction, and that, does that mean, like, just so I can understand what's happening, the blood pressure would lower? There'd be less of a blood pressure of a systolic blood pressure?
There would be a lower systole, absolutely. Okay. Yeah, because literally there's not as much to squeeze.
Okay, cool.
So the heart's only going to do what it needs to do. You know, it's only going to work with the pressures that it's got. So yes, you're absolutely right. When you've got leaky valves, one of the biggest giveaways is a much lower systole than should be there. Absolutely. Yep. Diastole's not affected, obviously, particularly, but the systole is. Yep. So it's actually a really good question, Laura. Like that sort of joins it all together, and it's excellent. Okay. So that's what happens with our leaky valves. We have a pressure problem. Now, this is not going to be on your test. You don't need to know it all. I put it there for your joy, interest. Having a think about what different valves, different places, the effect that it might have, or slightly different effects depending on where the valve happens to be. You don't have to answer this. This is literally something just for you to ponder on. So just to figure out the valve thing, what happens if my aortic valve is the one that's floppy? What's going to happen? Yep. What happens if it's my atrioventricular valves? What happens if they're floppy? Just have a think about it. Most important thing you know, though, is that blood's going to leak back into the chamber it came from, and that's going to change pressure gradients, which is going to change flow. Yep. Sometimes very, very badly. Okay. And just before we finish, I thought, what causes leaky heart valves? Because as soon as you get into pathophys, I swear everybody in a tutorial, including me, thinks we're going to die. We all go, oh, okay, there's leaky valves. I can get one of those. It's going to happen. Right. One that's big one, just most common actually, is congenital heart conditions. And a lot of people are born with one little leaky valve and they live with it quite happily for their entire lives, and it causes no problems at all. It's just a thing, right? You can hear it if you're actually listening to the heart, just a little flutter, but it doesn't do anything. It depends. It's really it's a scale thing. Right? So if your valve is really lazy and there's tons of backflow, then we've got a problem. If there's a teensy little bit, no, you'll be fine. Jess.
Can you hear a leaky heart valve on auscultation? Or is it just...
You can, depending on the amount. So if it's a really, like I was saying, if it's a really, really lazy valve, then yes, you can. If it's a tiny one, absolutely not. No,
okay.
Buckley's, yep. So enlarged hearts, anything that's going to cause hypertrophy, anything that's making the heart work too hard, obviously all muscles, all vessels are compromised when they're working beyond their capability. So that's a definite thing. Pulmonary hypertension, high blood pressure, obviously, the more pressure, the more effort, the more likely that organ is to not do well. And there are also complications after some infections, particularly any kind of infection of heart muscle or pericardium or anything like that. Okay. Very unlikely that you've got a leaking heart valve though, so please don't worry. I'm sure your doctor would have found it by now. Okay, however, we can do all sorts of different things on heart valve surgery. We can put clips on, we can put in an annuloplasty. There's 1,000 things that we can do, so don't worry. Even if you start leaking, you'll be fine. All right, that is it. All good. Are there any remaining worries, queries, questions? Looks to me like, as per usual, you guys are pretty much all over the blood stuff. It's fine. Yep. We will do some more pump and pipes next week, but mostly pipes. Right. So next week we're going to talk all about the blood vessels and the capillaries and all the wonderful things. Try and get your head around all of the pipes. There's just so many, but just the structure thereof and the function thereof, and we'll talk about that next week. Yeah. Cool. All right. I have to run off and give a lecture on pipes. So off I go to do that. And you guys have a wonderful afternoon. I will see you same bat time next week. And how many weeks have we got left? Where are we? So this is 7. So I think we've only got 2 more tutorials. Does that sound right to you guys? Yeah. Or is it 3?
It's sad. You're such a good teacher. I really
Except for last week when I went, I must remember to tell them about the vestibular apparatus. So I told you the wrong freaking thing. That was great. That was a stunning, stellar moment on my part. Well done, Kath. But, you know, like Taven said,
keep it fun.
she's human sometimes. She does say some very strange things. Always keep an eye on me. So those of you that actually picked it and put it on the discussion board, thank you. Good. Keep doing that, because occasionally I will say something that is absolute crap and you just need to pick me up. It's usually from tiredness or my brain's somewhere else, but yeah. All right. I'm going to lecture. I will see you all next week.
Thank you.
No worries.
Thank you, Kath.
See you guys. Bye.
Bye.